All right. Hello, everyone. My name is James Currie. I'm a master's student at the University of Alaska College of Fisheries and Ocean Sciences. And today I'm going to be talking to you about carbonate chemistry trends in Alaska nearshore environments. So to begin with, I'm going to give you all a quick overview of two of the ocean change consequences that this study focuses on. And the first of these is ocean acidification. So humans emitted 33.1 billion tons of carbon dioxide, or CO2, via energy production just in 2019. And the increase in CO2 in the atmosphere leads to a commensurate rise in CO2 concentrations in seawater. And as CO2 is absorbed by seawater, through a series of reactions, hydrogen ions are released. So it increases the concentration of hydrogen ions, it drives down the pH, or it acidifies the seawater, and it drives down the concentration of carbonate ions in seawater. And this decrease in carbonate ions makes it much more difficult for calcifying organisms such as clams and oysters and mussels to generate calcium carbonate minerals such as aragonite and calcite, which form their shells. So at the same time, marine heat waves, or thermally anomalous years, are expected to become increasingly common under future climate change predicted scenarios. And we've had a number of recent heat waves in the North Pacific, including one that took place between 2014 and 2016, famously known as the blob. And then we also had another thermally anomalous year in 2019. And so the issues of ocean acidification and warming are especially relevant to Alaska because the low temperatures of Alaskan waters makes them much more soluble to carbon dioxide. And at the same time, polar environments are expected to warm much more rapidly. So Alaskan waters are expected to both warm and acidify more rapidly than their temperate and tropical counterparts. And this has serious implications for a state where the fisheries are worth $4.6 billion and employ roughly 90,000 people. So despite the ocean change expectations for Alaskan waters and the negative implications of those changes for marine biota, there's a very poor understanding of nearshore carbonate chemistry fluctuations and trends within Alaskan nearshore ecosystems. So our study delves into these fluctuations in Kachemak Bay, which is this small bay that's attached to the much larger Cook Inlet. And it's this sort of nutrient-rich bay that's a bastion for marine fauna. And one of its dominant characteristics is the circular tidal-driven flow, where water flows in from the Alaska coastal current, up the southern shore, hooks around the top, and then exits via the northern shore back into Cook Inlet. Kachemak Bay is also bisected by Homer Spit and contains two eddies, one sort of at the, or what, two gyres, one at the head of the bay and one at the mouth. And the bay also experiences significant freshwater input from glaciers, stream runoff, and groundwater. So the objective of this project is to capture in high frequency resolution the carbonate chemistry fluctuations within Kachemak Bay and to determine which factors exert sort of the greatest control over these carbonate chemistry fluctuations. So within this study, we'll be comparing a heat wave year in 2019 to several non-heat wave years. And the parameters that we're focusing on are pH, temperature, dissolved oxygen, and salinity. And so to measure these parameters, we deployed sensor packages at 4 different locations in Kachemak Bay. And these sensor packages consisted of CFET sensors, which measure pH, HOBO loggers, which measure salinity, and mini-dot O2 sensors, which measure oxygen, dissolved oxygen. And these sensors were deployed 8 to 15 meters down. And we deployed them at 4 different sites, including one sensor deployed at Seldovia, or one sensor package deployed at Seldovia, one at Jackaloff Bay, one up on Homer Spit, and one in Bear Cove. And you might notice that these sensors are positioned in a way to kind of track that counterclockwise, tidally driven flow of seawater as it enters and exits Kachemak Bay. So at the same time, whenever we deployed these sensor packages or went to service them, we collected seawater samples via Niskin bottles. And then in the lab, we processed these water samples to determine the pH, total alkalinity, and salinity. And we could then apply these seawater standards to the sensor time series to calibrate the time series and to adjust for any sensor drift. So we've collected the time series and we've just begun to start working on them. So we have a very cursory overview of our preliminary results, but within these, we can begin to see seasonal, annual, and interannual fluctuations in seawater chemistry. And we'll be working through these data over the coming months in order to reduce the uncertainty. And so here we have our Homer time series, and you can kind of see we were hampered a little bit by COVID restrictions and thus weren't able to get to the sensors for servicing and validation as often as we'd hoped or planned. But despite that, even this preliminary time series, you can see or you can begin to see how seasonal variability in pH differs at this site from year to year during the winter-spring transition. So we have 2019 up here and 2020 down here. And over this period from January 1st through April 30th, both years, we can see large differences in pH variability over the course of the winter into the spring transition in 2019, and much smaller changes in sort of the pH variability over the course of the season in 2020. So we see much higher variability in 2019. And at the same time at this site, we also observed statistically significant differences between the temperatures in 2019 versus 2020, which you might expect given the heat wave of 2019. Interestingly, thermodynamically, we'd also expect that as water temperatures started to increase, we would see a corresponding drop in pH. And that's actually what we observed in 2019, just visually. We also observed much larger ranges in oxygen concentrations in the spring and early summer of 2020, which is the blue here, as opposed to 2019, which is the red here. So these ranges are really different from in 2020 versus 2019. We see a 5.16 fold increase in the range of O2 concentrations on average over this time period in 2020 over 2019. So that's kind of a pretty serious jump. And then in Bear Cove, with the 2 years that we have to compare, we can observe agreement between the pH signals as we transition from wintertime into spring. And this is unlike what we observed in Homer, where we saw much higher sort of interannual variability. There's more agreement here between the overall pH interannual variabilities, and that's borne out in the standard deviations. It should also, I'd also like to point out that the pH, the peak pH values, which we associate with the sort of the peak of photosynthetic activity, actually occurs over the course of these 2 years, so 2020 and 21, within 2 days of one another, which is kind of an interesting pattern. So in the future, the next steps of this study will be to tease out in much greater detail the temporal and regional differences within Kachemak Bay and these different sort of sensor locations and different times. We're also going to extrapolate the carbonate system using the seawater parameters that we've measured. And from that, we can use the PCO2 to determine CO2 flux into and out of seawater in these different bodies of water over different quantities of time. We also may be able to detect water mass movement. This is a figure produced by the Kelly lab, which shows how you can use the sort of unique signature of temperature and salinity in individual water masses to track them as they move through Kachemak Bay. So maybe we'll be able to see that. And we should also be able to calculate aragonite saturation and sort of when and where are most environmentally suitable for calcifiers. So Homer seems to experience disparate pH variability during the winter into spring transition, and the oxygen concentrations there also show very high interannual variability within the spring and into the summer transition, whereas Bear Cove is much more consistent during the same sort of rough seasonal time period. So with that, I'd like to thank my advisor, Dr. Kelly, my committee members, Dr. Minks, Dr. Miller, and Dr. Burt, everybody else in the Kelly lab, and everyone else who's helped us with this project. Thank you all very much, and please feel free to reach out if you have any questions.